Hi, everyone. So this is Professor Kwok from City University of Hong Kong. So today we are going to talk about building and innovating DNA. So in today's talk, we will have four sections. So the outline is shown here. So we'll talk about DNA for data storage as an aptamer, nanostructure, and also DNA for drug delivery. So we'll first revise the differences between DNA and RNA and the history of discovering of the discovery of DNA double helix. So DNA is a double helix shown in here. And DNA, as many of you know, carries genetic information. And a gene is a DNA segment that encodes functions and is considered a basic unit of inheritance. DNA is made up of single unit called a nucleotide. It has a sugar here, also the phosphate backbone, which is not shown on this diagram. It has four bases, the A, G, C, and T for DNA. If it's RNA, then it's the U. So DNA is using deoxyribose shown here, and RNA is using the ribose shown in here. And then A will pair with T, and then G will pair with C, or A can also, in RNA, can pair with U. And these bases are interacting by this hydrogen bonding, base stacking, and etc. So a single DNA molecule can be of millions of nucleotide long in length, which condense into chromosome. So in human, we have around 3 billion base pairs. So here are the key players for the discovery of the DNA structure. So on the left-hand side, we have James Watson and Francis Crick. In the middle, we have Rosalind Franklin, who took this famous photograph 51. So they were working on the answer at the same time, but in different university in England. So photograph 51 was taken by Rosalind Franklin in her lab that support the double helical nature of DNA. So without her consent, the photo was taken from the lab by her Maurice Wilkins, and he passed on to the Watson Crick, which helped them to finalize their proposed model. And they have published a paper in 1953 showcasing the double helical structure of DNA. So of course, James Watson and Francis Crick get the Nobel Prize. Unfortunately, Rosalind Franklin passed away in 1958 or 1959, so she wasn't awarded the Nobel Prize in 1962. So there's this video that you can find in TED, which talk about Rosalind Franklin, the unsung hero of DNA. So this, which gives you the more historical perspective of the DNA discovery story. So you can find the video by typing the words in here in YouTube or other channel. So as you all know now, so DNA is the blueprint of life. And so next we'll talk about how we can actually use this DNA as a material for information storage. So DNA blueprint can be broken down into these three parts. So it codes for it. So DNA encodes instruction for building and maintaining living organism. And then it will code for the protein. So this is most important information encoded by DNA is the protein synthesis. And also you can scale. So DNA inside a single cell contains 20,000 genes, and there are trillions of cells in the human body with more than 200 functions. And then there's this genetic variation. So everyone's DNA is slightly different. So this comes to make each one unique, and that's why you will see different types of people, and then they have some similarity and some differences. For example, in terms of the height, in terms of intelligence, in terms of gender, and etc. And genetic variation can arise from mutation. So this is something that is stored inside your DNA. But that doesn't mean you cannot adjust. I mean, like there's also this term called epigenetic, where you can interact with the environment to change your phenotype. So which will not be covered in this talk, but you can feel free to explore more yourself. So for those who are curious, so this is also a very interesting book, and students at your age can already read it and understand it. So it will tell you how our life and ourselves are controlled by genes, and how genes determine if we are altruistic or not, why and why not it is beneficial to evolution also. So mutations, so how antibiotic resistance happens is that, so there are a lot of germs and some are drug resistant when we treat them with antibiotics. So antibiotics will kill the bacteria causing the illness, as well as the good bacteria protecting the body from infection. And then the ones that survive the antibiotic treatment, which is also called a drug-resistant bacteria, is now able to grow and take over the colony. And some bacteria will start to give rise to drug resistant to other bacteria as well. So this is how mutation could happen in bacteria. There's also this sickle cell anemia. So this is a very common example of a mutation and will cause malfunction of the hemoglobin protein folding, which reduce oxygen absorption in the red blood cell. So however, having a sickle cell trait provide protection from malaria from many studies. So also this red-green color blindness is another sex-linked or X chromosome mutation. So for normal people, they will see this picture. For the people with red-green color blindness, this is what they will see. So as DNA can be used as a genetic information carrier, many scientists have also thought about maybe DNA can be used as a data storage. So in 1994, so there's a problem of a truffle salesman can be solved using DNA as a substrate. So this is the picture. You can think about and read more about it later on yourself. In 1996, a visual icon was converted to DNA and stored in bacteria. So DNA is preferred over other molecules because of its predictable and specific base pairing. So you can convert this DNA sequence into this binary code of the image, or vice versa. So what's the advantage, or what's the background about DNA as storage? So first is this capacity. So 1 gram of DNA equals to many data in the computer unit. It's also very stable, so it lasts 1,000 times longer than silicon-based storage media. Also, the size can be stretched to 2 meters and only 4 microns in diameter. It can be used as a backup, so rapidly replicated by polymerase chain reaction. The data type is ATGC from DNA, and computer binaries is 0 to 1, 0 and 1. So there's this Microsoft team that has developed this using automated DNA data storage. So again, you can type in these keywords in YouTube, then you will be able to find and watch the video. So molecular storage using DNA. So it's very simple. So you can write or synthesize. So you can train the algorithm, bind strings of bytes into DNA. So you can store it. So in vitro, it could be frozen solution or dry solid. In vivo, it could be a live cell. And then you can retrieve the data. So it's random access. And then once you retrieve the data, you can read it. So then decode DNA back to the original data. So in theory, you can use DNA to store information. So you can also look into here. So this is the Watson-Crick base pairing. And then you can have different mechanism to store the data, and you can incorporate total displacement, DNA circuit, neural networks, or compartmentalized circuit. So these are different ways to store data and to convert it into different signal. And then just like the computer, you can convert it into digital file. They can be stored or they can be read using this binary code. They can also convert then back and forth to DNA sequence. And DNA sequence, as mentioned, can be synthesized into DNA molecules, and then it will operate the file in this way. So the potential problem for using DNA is the loss of information. Although DNA is stable, it can still have accident degrading of information in there. If there's an by enzyme presence in the environment that can have severe consequences. So TNA is a spherules where it's a different type of sugar in the subunit of the nucleic acid. So it's biologically stable alternative. It's a next generation information storage. So artificial polymer, they are not susceptible to nucleus degradation. So LDNA is another type of DNA molecule. So this is the stereoisomer of the natural deformed DNA. So you can also use that to store information. So this is an experimental study demonstrating how LDNA evade biodegradation as an isomer. So LDNA in principle possesses the same information capacity as the natural deformed DNA. It's resistant to degradation and can serve as information storage. And Professor Zhu Ting at Westlake University have synthesized a 90 kilodalton DNA polymerase that allows the replication of the LDNA. And it also potentially can allow for the assembly of kilobase size mirror genes. So this is an example of company working on DNA storage. Currently, they work on creating their own DNA-based computing platform. And the objective is to cut the cost of information retrieval even from the oldest archives, and to store information without having to build stadium-sized computer center. So more than $9.3 million investment was collected from this startup. So you can also check their website later on. So using DNA or RNA for different application. So one of them is called aptamer. So aptamer is a short single-strand RNA DNA with high target selectivity. They are also known as the chemical antibody. So as you can see in here, there's a sequence, and then it can fold into different structure by ATGC base pairing. And then this structure can help to recognize the target or the biomarker shown in this case. There's also videos that you can watch that will help you to understand more. So you can again find it in YouTube. So aptamer can bind to a variety of targets, including metal ion, small organic molecule, such as amino acid, ATP, antibiotics, protein, and even whole cell microorganism. So application includes diagnostic and therapy, for example, cancer therapy. And compared to other biosensors, they are highly chemical stable. So this is a short summary table comparing the aptamer versus the antibody shown in here. So it's much smaller. The generation of aptamer usually is much faster than generating antibody, and for different other comparison, you can look into this table later on. So how it works? So aptamer can work in the following way. So it will first compete with small molecule or protein to inhibit the target. Can have antiviral property, can act as a carrier for drug as well. So first, aptamer-based therapeutic is called a Macogen. So it's a modified DNA/RNA aptamer. So it's against the vascular endothelial growth by blocking the VEGF gene. So this will help to make the aptamer work. So it's approved by FDA, and it is a age-related macular degeneration. So this can help to solve the problem. So there's this newest FDA-approved aptamer. Name is shown in here. So it's used for the treatment of this GA secondary to age-related macular degeneration. It's discovered at this company, again approved by FDA in 2023. The aptamer itself is very 9-nucleotide aptamer attached to polyethylene glycol. The function is that it will inhibit the complement protein C5. And so you can read more by typing the name of the aptamer or the drug, so you will understand more. So for aptamer future, so one of the current limitation is the problem with the target-specific delivery. So you can basically have the aptamer and tether into a different carrier for the delivery. So for example, if you want to deliver into the mouse, then you can put on this cargo and carrier. And then if you have a specific organ that you're interested, then you can put in the right organ-specific carrier, then you can bring the aptamer, or you can bring the drug to the target organ, or even to the target cell or target compartments inside the cell as well. So third is the DNA as nanostructure. So basically, DNA can act as a building block. It can be 1 dimension, 2 dimension, and 3 dimension, shown in here. The sequence is designed by computer-based program, and it can perform the self-assembly. Once you have the sequence in place, then it can self-assemble. Base pairing is specific, ATGC pair. So forms a double-stranded DNA because of the design, for example, shown in this nanostructure here. This is a tetrahedral DNA nanostructure. It has a unique geometry, so you can design it to be like a star, to be different shapes. You can also make very complex structure out of the design of DNA sequence. Folding into a particular structure will require the scaffold and scraples and heating with salt. So you need the DNA, you need different strands. So each color here is a different strand of DNA, so they can assemble together to form this unique nanostructure. So you can use it to apply for biosensing, bioimaging, and biomedical application. So target will include nucleic acid, enzyme and proteins, pH, small molecule. So probe immobilized on the surface will hybridize with the target and produce a distinguishable signal. So shown on the diagram on the right here. So bioimaging usually will use the fluorescent. So you can use fluorescent imaging, bioimaging with this DNA nanostructure. So another application is cell assembling and capturing. So you can have it apply into a different field, not only in biosensing, but also use it for cell biology or cancer biology. So it's easy to design the program DNA to make into different shapes of the nanostructure shown in here. It's biocompatible, it's stable. However, you need to have special care because this structure require design and conditions that are specific for them to form. So they're pH and temperature dependent, for example, and other chemicals might dissolve DNA as well. So in the future, new computer and production methods are required to have a better design. There's a limited number of scaffolds that exist. So there's also active research ongoing to see what kind of other structure it can be designed. And sequence has to be optimized during the design process. So with some fine tuning is needed to make into the nice structure shown in here. So the last part is a DNA for drug delivery. So the advantage for using DNA for drug delivery is that it's biocompatibility, control of drug release size, and development of nanonet medicine. So here are some examples. So where you can again build into different nanostructure, and then they can interact with the drug. And then when it goes inside the mouse or the cell, then it can release the drug, and then it can be treated, for example, for cancer. So the nanostructure DNA can be used as a vehicle holding the drug for drug delivery. So such method has high affinity due to specific binding of aptamer and involve the bonds between the ligand and the receptor. So a general method, so a target of organ, so that's the primary targeting. Endocytosis, a ligand receptor recognition, secondary binding, targeting. And then the third one is to bring efficient immune response. So then this will help to kill the cell or to exert the function of the drug. You can also use DNA as a sensor. So basically you can immobilize them on a surface, and then when the target binds, then you can trigger a signal change. It can be electrochemical, it can be fluorescent, it can be visible. So biosensor is a device that measures biochemical reaction by signaling. So the first biosensor was developed for oxygen detection in 1965. Again, as mentioned before, target can be nucleic acid, protein, metal ion, and small molecule. So then you can imagine can have a very wide range of applications, for example, medical, environmental, forensic, and for safety. So it has very high affinity and high selectivity by developing this. and future might involve cheaper, faster, and improved biochemical assays. However, practical analysis technology is limited, and there are limitations with tuning the probe as well. So this, again, is an active research area to improve how we can use DNA to build biosensor. So the take-home message for this short lecture is that, so besides understanding DNA as a genetic information carrier, we'll cover a few topics, DNA for data storage, DNA for drug delivery, aptamer, DNA as nanostructure, and etc. So all in all, so DNA functions are way beyond just storing genetic information. We can play around with the DNA for a lot of different applications. For example, it's a potential therapeutic tool which presents numerous advantages against currently used treatment. For example, antibody or small molecule. So also its chemical properties allow us to modify its structure and create new shapes. For example, we can use modified nucleic acid, the TNA or LDNA. We can also design, with careful design, we can make it into tetrahedral and other nanostructure. However, there are still quite a lot of techniques and technology needs to be developed to overcome many of the hurdles that I present earlier today. And of course, you can read more or you can ask AI to gather more information if you're interested in these topics. DNA is an amazing molecule from understanding the structure and how we use this structure to build to different tools and different vehicles for different applications. So in the future, I hope all of you will play a role or do some research or do some study with DNA. So last, this is my group. This picture was taken maybe a few years ago. So I'm Professor Bob from Department of Chemistry at City University. So my interest is on nucleic acid, particularly on RNA and aptamer and G-quadruplex, which is a very special DNA and RNA secondary structure. So with that, I'd like to thank you for your listening. If you have any question, feel free to email me. And with that, I will end the talk here.